Hello, everyone. My name is Lucas Zhang, and I'm a rising senior at the Illinois Math and Science Academy. And today, I'd like to share a new perspective on a devastating disease, amyotrophic lateral sclerosis, or commonly known as ALS, and why the gut-brain immune axis may hold the key to future treatments. As I said before, here's a bit about me. I'm a rising senior from Illinois Math and Science Academy. I participate in events such as IYNA, HOSA, swimming, and bodybuilding, and I plan to go into some sort of specialized surgery, possibly cardiac or neurosurgery. So, first, for some background on ALS, what exactly is it? So, ALS is a progressive neurodegenerative disease that affects motor neurons, the nerve cells responsible for controlling voluntary muscles. Over time, patients will lose the ability to move, speak, eat, and eventually to even breathe. The average survival time after diagnosis is just 3 to 5 years. ALS is a global issue as it affects over 200,000 people every year. And as this chart from Nature shows, that number is projected to rise significantly by 2040. Despite decades of research, there's still no known cure. Traditionally, ALS research has focused on genetic mutations and neural mechanisms. But that view might be too narrow. What if we zoomed out and looked at the whole body and not just the brain? This is what I'll be talking about today, including the gut microbiome and immune system. I'm going to go over the gut-brain immune axis. It consists of three systems that constantly communicate with each other: the gut microbiome, the immune system, and the central nervous system. Disruptions in one can influence the others, and emerging research has shown that changes in gut bacteria may actually influence neurodegeneration, as is seen in this diagram here. As for existing research and outlining the problem, ALS has been found to be a progressive motor neuron disease with no cure, as of now, and a median survival of 3 to 5 years. Most traditionally, research has focused on genetics and the neurons themselves, trying to understand what goes wrong inside the brain and spinal cord. But in recent years, that focus has begun to shift. Emerging evidence reveals a role for the gut microbiome in neurodegeneration, and which has been explored in other prevalent neurodegenerative diseases like Alzheimer's or Parkinson's. However, these interventions, like FMT, have not been specifically tested in ALS-specific trials. This gap is exactly where our work comes in, proposing a systems-level framework to better understand and potentially intervene in ALS progression. So, for the purpose of this presentation, I plan to introduce a novel perspective on ALS, the gut-brain immune axis, present evidence from both humans and mice that gut imbalance accelerates ALS, and explore how restoring microbiome health might slow progression of the disease. So, as for our research hypothesis, we propose that gut dysbiosis in ALS causes barrier breakdown, which allows toxic metabolites into circulation, chronic activation of immune cells like microglia and T cells, and reduced production of GABA and nicotinamide, which worsens neuronal stress. In our paper, we specifically identify a triple failure: loss of microbial species, loss of metabolites, and loss of barrier integrity as a key factor in ALS pathology. So, our proposed method of intervention is fecal microbiota transplantation, or FMT. We plan to use the SOD1G93A ALS mouse model. Mice that received FMT from healthy donors showed improved motor function, lower inflammation, and restored gut composition. In contrast, mice treated with antibiotics or raised germ-free had worse outcomes, highlighting how important a balanced microbiome is. We monitored motor skills, inflammation, and microbial composition in order to verify the potential effects of this treatment. To start, we'll talk about microbiota disruption. ALS patients and ALS mice lose key microbes early. Akkermansia muciniphila, which supports the mucus barrier and produces nicotinamide, Bifidobacterium and Lactobacillus, which produce GABA and SCFAs like butyrate. Without these, the gut becomes permeable, neuroprotective signals drop, and inflammatory cascades are activated, even before symptoms begin. Next, we'll talk about the leaky gut and inflammation. This leaky gut allows LPS, a bacterial toxin, to enter the bloodstream. It's detected at higher levels in ALS patients and models. LPS activates TLR4 receptors on immune cells, including brain microglia, shifting them into a pro-inflammatory M1 state, which contributes directly to neuron damage. This is one of the clearest examples of how gut dysfunction can spark CNS inflammation. Next, we'll talk about the loss of neuroprotective metabolites, which includes decreased SCFAs, GABA, and nicotinamide. SCFAs regulate anti-inflammatory microglia, GABA reduces cortical hyperexcitability, and nicotinamide supports mitochondrial repair. In one experiment, restoring nicotinamide via Akkermansia supplementation delayed symptom onset in mice, which shows a positive effect. Finally, immune and neural impact. ALS patients show a Th17 TREG imbalance favoring inflammation, elevated cytokines like IL-17A and IL-6, and hyperactive microglia that harm motor neurons. FMT, or targeted probiotic therapy, can reverse some of these effects in mice, restoring IL-10, reducing IL-17A, and shifting microglia toward a homeostatic state. To conclude, ALS is not just a neurological disease. It involves systemic inflammation and gut dysbiosis. Disrupted microbiota and barrier function trigger immune responses that ultimately harm neurons. Restoring balance through targeted microbiome therapies could represent a new frontier in ALS treatment. This is a call for the research community to consider the whole body systems involved, and not just the symptoms at the surface. Here are some of our references, and I'd like to acknowledge my fellow researchers Nagamuda and Pooja Sempeli, as well as my mentor, Dr. Ashwin Mohan. Thank you all for listening, and I hope you have a great rest of your night. If needed, here are ways to contact me in order to further discuss our research.